Hello, today we'll be going over neurological disorders associated with GNAO1 variant V202I. It's just an introduction to our work and poster here. We'll be looking at a variant of the alpha subunit of G protein O. This variant is a change from valine to isoleucine at amino acid number 202 and has been found in a patient experiencing symptoms of neurological disorders, including ADHD, autism, and some difficulty with speech. To analyze what was going on here, we collected a variety of bioinformatical data on not just this code variant, but also variants R209C and 209H. These variants are important because they were found to be associated with neurological disease and are in close proximity to V202I. In general, G proteins are essential for cellular function. They act as switches that mediate the activation and inhibition of cellular processes, including the production of cyclical AMP and the activity of ion channels. So because of this, G proteins do play a major role in the proper function of neuron cells. So looking at our very first figure here, we have an alignment of the common variant as well as the code variant. As you can see at nucleotide number 604, you have the guanine highlighted in gray. It's going to get changed by a single point mutation to an adenine shown in the red. And this is going to change the amino acid from a valine to an isoleucine. So in figure 2, we have two different alignments. In A, we have homologous protein sequences of different proteins in the G protein family. And in B, we have alignment of different paralogs of the same GNAO1 and found in different species. So as you can see in figure 2A, from around the 202 region shown in the red valine all the way over to the 209, that region seems to be very highly conserved. And in B, we can see that the entire region that we looked at was completely conserved across different species. Moving on to figure 3, we have a box diagram showing the GNAO1 sequence. So from amino acid number 32 to 354, that is our G alpha domain. In A, the pink line is our V202I. And right next to it is our next two variants, R209C and 209H. And the point of this is to show you just how close together V202I is to R209C and R209H, which have been known to cause neurological issues.
As we see in figure 4, string was used to illustrate protein-protein interactions within the G protein pathway, and the proteins were labeled by their corresponding functions. G proteins interact further with G protein coupled receptors, adenylate cyclases, and ion channels. Furthermore, signaling between GNAO1 and GNAI1 was discovered using an assay in vivo. Figure 5 depicts a scatter plot of CAD scores of known GNAO1 variants. This method is usually used for scoring the deleteriousness of single nucleotide variants, as well as insertion-deletion variants in the human genome. The 175 to 275 region, where our variants are located, shows an increase in the frequency of deleterious mutations. As for figure 6, a 3D model was constructed by using ISARA to depict the different variants of GNAO1 as seen in 6A. The comparison between the different 3D models shows several structural changes between the variants and the wild type as seen in 6B. In figure 7, the RMSD analysis and the alpha carbon RMSDs of GNAO1 residues are presented as line graphs. The region of interest between position 200 and 225 shows little variations.
All right, so in figure 8, what we're looking at are a few different tables of these term comparisons of proteins, which were all found doing searches in the Gene Ontology and KEGG Pathways databases. And these proteins were found to repeatedly have similar protein functions as the GNAO1 protein, which could mean a relation to how they all affect patients with these disorders and how they're passed down through genetics. So in conclusion, we believe that the mutations in the G-alpha subunit in the amino acid range of 200 to 250 appear to be correlated to neurodevelopmental disorders and neurological diseases. Furthermore, we believe that the mutation of the GNAO1 coding sequence to be a suspect of the patient's diseases and symptoms. However, we can't at this time determine how the mutations are causing the neurological disorders or why this region of the alpha subunit is critical to function. And potentially additional runs, particularly of the R209C and R209H regions, need to be done to confirm the alternation in protein movement.